about the Hardy-Weinberg equation and how we actually use it to calculate allele frequencies and interpret whether or not evolution has occurred. Most often, I'm going to ask you simply to calculate allele frequencies and genotype frequencies. But the purpose of this is to be able to quantify evolution. So you can look at a population at one point in time, calculate allele and genotype frequencies, and then calculate allele and genotype frequencies after a suspected evolutionary or selection event to see if evolution has actually occurred within a population. So the Hardy-Weinberg equation makes some assumptions. Hardy-Weinberg works because it assumes at a particular point in time there is no evolution occurring. And if no evolution is occurring, we make some assumptions about the impacts of certain factors on that population. So first we have to assume that no mutations have occurred for the allele in question. So if no mutation has occurred, evolution is not going to occur at that particular allele. We also assume that there's no differential survival, which means there's no natural selection going on. No individuals are surviving at a better rate because they have a particular version of one of these alleles. We also assume that there's random mating, meaning that individuals are selecting from a large population of mates and not choosing that allele in that specific version of the allele in their mate. We assume that there's no genetic drift. This means no chance fluctuations in allele frequencies from one generation to the other. This is also called large population size. So what this means is that the population is sufficiently large enough that those chance events are not affecting allele frequencies. So genetic drift can be violated very easily in a small population because chance events are going to affect that small population potentially much more than a larger one. And finally, we have no gene flow. This means that there's no alleles coming in or coming out, going out of the population. So Hardy-Weinberg equation is based off of the fact that that all of the frequencies of all of the allele frequencies of a particular gene are going to equal 1. So we're going to be working in decimals here. So this means that the frequency of one allele plus frequency of another is equal to 1. Now we get the Hardy-Weinberg equation by doing a binomial expansion of p plus q equals 1. That means multiplying p plus q times p plus q. Now we're not going to do that. I don't expect you to do that. You don't even necessarily have to know how to do that. But to get there to the Hardy-Weinberg equation, we're going to multiply those two. Now if you recall from lecture, each of these parts of the equation are equal to some information in the population. So p squared is going to be equal to your homozygous dominant individuals in the population, so individuals with homozygous dominant genotype and therefore dominant phenotype. 2pq is going to be equal to your heterozygous individuals in a population, which is going to also be equivalent to the dominant allele frequency. Dominant phenotype. So these two together, p squared and 2pq, are going to represent our dominant phenotype individuals. Our book uses an example where 2pq is actually represented by an incomplete dominant situation. So you can actually see the individuals that are heterozygous, which is really helpful in learning this equation. Finally, q squared, which is the one we're going to use the most, is going to be equal to the individuals who are homozygous recessive and therefore have the recessive phenotype. Now, this is really useful information because if you know the recessive phenotype, you also know the recessive genotype, and that's going to be the basis or the starting point for a lot of our equations. Now, all of this is equal to 1 because this is a frequency. So when we do this, use this Hardy-Weinberg equation, we're going to follow a set of specific steps. The first one, and the most important one for getting you started off correctly, is to determine if the problem gives you an allele frequency or a genotype frequency. And you have to read the problem carefully and decipher what is going on. I'll give you a hint. if it's giving you an allele frequency, it probably is going to say the allele frequency is, etc. If you are being given a phenotype or a genotype frequency, then you're given slightly different information, and you're going to most likely start with Q squared. So you have to think about what they're telling you. If they're telling you a phenotype, they're giving you a genotype frequency of some sort. Next, you need to calculate your allele frequencies based on what you're given. So you might be given Q squared, and you have to calculate Q, and then P, or you might be given Q, and then you calculate P. Next step, calculate whatever genotype frequencies are needed for your question, and then answer your specific question.